respected dignitaries and the incredible guests. I'm so grateful to welcome all the in International E-Conference. I, Dr. Bharti Wadekar, Assistant Professor, Microbiology Department, ZSCT Thakur Shyamnarayan Degree College, Mumbai. It gives me immense pleasure to welcome you for this International E-Conference, which is jointly organized by the IQAC and Microbiology Department of ZSCT Thakur Shyamnarayan Degree College, Kandivali East, Mumbai, Tilak Maharashtra Vidyapeeth, Pune, and Microbiologist Society, India. I feel privileged to open this event and stand here where, where many accomplished people who I admire and look up to once stood. A very special welcome to Honorable Dr. Tilak, officiating Vice Chancellor, Tilak Maharashtra Vidyapeeth, Chief Guest Dr. Hemant Tripathi, Research Fellow, Africa, and Dr. Pratik Dhawan, Postdoctoral Research Associate, California, US. I express my gratitude to our esteemed principal, Dr. Sharat Fulari, sir, for their for their approval. They have assisted us in not just our development but also in enhancing our creativity and knowledge. And last but not the least, a heartfelt thanks to our esteemed lecturers, without whom would be the incomplete the event. They were always here to assist and advise us in unimaginable ways. They help us with the development of ideas in research and the execution of those ideas. It's my pleasure to walk you through the conference, introduce you to the organizing institutes, speakers, and delegates that have come from all over the country today to share their knowledge and enhance our mental horizon. So it's warm welcome to each of you who is participating in this international conference. First college, ZSCT Thakur Shyamnarayan Degree College, Mumbai. Mumbai was the most populous city in India with an esteemed city proper populations of 1.25 crore, living under the municipal corporation of Greater Mumbai. It has the highest number of millionaire and billionaire among all the cities in India. Mumbai is home to the three UNESCO World Heritage Sites, the Elephanta Caves, Chhatrapati Shivaji Maharaj Terminus, and the city distinctive symbols of Victorian and the Art Deco building designed in the 19th and the 20th century. Everyone knows about Mumbai. It is the capital of Maharashtra and economic center of India. There are the 15 top rated tourist attractions in Mumbai: Gateway of India, Sunset of Marine Drive, Bandra Worli Sea Link, Taj Mahal Palace, pictures of the Taj Mahal Palace, Mumbai, Kanheri Cave, Chhatrapati Shivaji Maharaj Vastu Sangrahalaya Museum, Global Vipassana Pagoda, etc.
Mumbai, also called as the City of Dreams, is truly a place to visit in India. From the best nightlife ever that will give you the run for your money to the most elite places possible, Mumbai won't disappoint you in any aspect. With continuous influx of people from across the globe, it is now a home to various cultures and religions. One of the best places to visit in Mumbai is Gateway of India. Built in the 20th century to commemorate the visit of King George V and Queen Mary to Mumbai. It is a large arch with a height of 26 meters and is done in Indo-Sasanic style. However, if one looks closely, traces of Muslim architecture styles can also be traced. It is an ideal photographic destination. Second, Marine Drive. Marine Drive is the heart of the city. It is a place where people experience the eternal beauty of the city. Marine Drive is also referred to as Queen's Necklace because of the street lights that make the road look like a string of pearls and create an illusion of a necklace. Third, Elephanta Caves. The caves are said to be one of the oldest rock-cut structures in the country and is a perfect example of archaic Indian art associated with Lord Shiva, depicting him as the destroyer, creator, and preserver of this universe. This is also a UNESCO World Heritage Site. Fourth, Juhu Beach. Juhu is one of the largest and most visited beach, home to so many celebrities that it has become the ideal destination for sightseeing. Beaches come to life in the evening when people start enjoying themselves in the beauty of the place. It is also popular for lip-smacking street food. At number 5th, Bandra Worli Sea Link. The Bandra Worli Sea Link, which is also called as the Rajiv Gandhi Sagar Setu, is situated on the Worli Sea face. It interfaces the island of Worli to Bandra over the Arabian Sea. Do not forget to check out this place during monsoons, as it will give you mesmerizing scenes to capture for a lifetime. Sixth, Siddhivinayak Temple. It's a Hindu temple dedicated to Lord Sri Ganesh. It is a standing demonstration of beauty and architecture. It is very famous in not just Mumbai but in various parts of the country. Situated in Dadar, it is one of the most beautiful places to visit in the city. 7th, Haji Ali Dargah. Haji Ali Dargah is a renowned place for Muslims. It is a whitewashed structure that is spread in the area of 4,500 meters and is surrounded by white pillars. It is situated in the middle of the sea that is attached to the part of the land which gets submerged in water in the events of high tides. This place is a holy shrine for Muslims but has attracted people belonging to different religions. 8th, Essel World. Essel World is the biggest amusement park in the city and is a huge tourist attraction. It is situated on the coastal Goregaon town in northwest Mumbai. People from all age group visit the island of fun and frolic and have fun, stimulation and immaculate satisfaction. 9th, Mount Mary Church. Mount Mary is an ancient church that was built in 1640 and was later rebuilt in 1761. This church is situated on top of a hill that is known as Sundarban Bandra, which looks like the Arabian Sea. Church is believed to have healing powers, and that is the reason it is always swarmed with devotees from all over the globe. 10th, Colaba Causeway Market. It is an ideal place for shopping in Mumbai. It is a street market that provides a range of things to shop and is situated in Colaba area, which is always brimmed with tourists. You can buy amazing stuff here at cheap prices if you have great bargaining skills. 11th, Film City. Film City is a major highlight in the city of Mumbai, as it is home for Bollywood and many people always desire to see the real life for real ones. It is located near Sanjay Gandhi National Park in the vicinity of Airy Colony in Goregaon. 12th, Versova Beach. It is situated at a distance of 11 kilometers from Mumbai. Fishing is considered to be their main source of livelihood. The local residents of this area are referred to as coolies. You can even go for horse and donkey rides if you wish to, and don't forget to take a glimpse of the coolies catching fishes. It is a sight to remember. 13th, Sanjay Gandhi National Park. Sanjay Gandhi National Park is a wildlife reserve in Mumbai. It is spread over an area of 104 kilometers and is very popular among wildlife and nature lovers. This park is located in the northern span of Thane city. 14th, Mahalakshmi Temple. This temple is built to honor goddess Mahalakshmi. It is most crowded on Fridays and also during Navratri festival. Thousands of devotees visit this shrine to seek blessings from the goddess.
Second organizing body is Tilak Maharashtra University, Pune. Pune is formerly known as Pune. It is the second largest city in the state of Maharashtra. Is widely regarded as the second major IT hub of India and the top automobile and manufacturing hub of India. It is known as the Oxford of the East with the presence of a wide range of educational institutions, It also has a rich cultural and spiritual history with many temples and ashrams spread across the city. In addition to local adventure and history junkies, Pune also attracts the many international tourists because of the Oso International Meditation Resort that is located in the heart of Pune at Koregaon Park.
Pune, also called the Oxford of the Street, is a glorified place which is located near Mumbai. The ancient history of this city dates back to 1600 years and can be depicted through the old settlements. Famous for its colleges and the serene atmosphere, this place is an apt destination for weekend getaways from Mumbai. Number 1. Dawa Dushit Halwai Ganesh Temple. This temple was built by a Halwai in remembrance of his lost child. Thousands of pilgrims come here for their wish to get fulfilled. The temple is dedicated to Ganesh and is built with white marble. Second, Darshan Museum. This museum has light and sound shows shown through 3D holographic technologies. The shows are based on the life of Sadhu Vaswani. The story depicts the moral lesson of universal wisdom and encompasses different life stories of the saint. It is a 100-minute show showing everything about him. Third, Shaniwar Wada. The place used to be the home to the Peshwa rulers. It was built by Shivaji in 1736. Now, the palace has been destroyed due to the fire which broke out in an ancient time, but still it holds an important place in the city. At number 4, Srinagar Fort. It is one of the isolated cliffs in the Bulaswar range of the Sahyadris of the city. The fort holds many myths and tales which are associated with the building of the fort. Today, the place stands as a great example of a glorious period of Pune and comes under some of the best tourist places to visit in Pune. Fifth, Pula Deshpande Garden. Considered to be one of the largest gardens in the city, it is also called as Pune Okyama Friendship Garden. The name was coined due to the 300-year-old Okyama Koraku in garden. The most beautiful thing to watch is the uncanny natural flow of water. The garden spread across an area of 10 acres. Sixth, Chaturshringi Temple. The temple was built during the reign of Chhatrapati Shivaji Maharaj. The tale of the temple goes around a merchant named Dulabh Seth Pitambar Das, who was a devotee of goddess Chaturshringi and wanted to visit all the temples dedicated to the goddess. But as he grew old, he had some problem in commuting, and thus one night the goddess appeared in his dream and told him to dig at a specific place. That's where the temple today stands. At number 7, Raja Dinkar Kelan Museum. This museum has been built in Rajasthani style, which is away from the hustle and bustle of city life. The museum displays a unique collection of artifacts and Indian arts and crafts. The museum has 36 sections with walls being carved with specific traditional paintings. 8th, National War Museum. The idea was first laid in 1996 and was sponsored by Express Citizens Forum. They set up a war memorial fund and asked the citizens of the city to contribute in it. The memorial is 25-foot stone pillow. 9th, Parvati Hill. The hill gives you a beautiful and mesmerizing view of the city. It has 108 narrow steps which lead to the temple of Balaji Bajirao. This was once the private shrine of the Peshwas and was the final resting place of Nana Sahib Peshwa. It has a Parvati museum which shows the portraits of the Peshwa rulers and the manuscripts with pole points of that era. 10th, Aga Khan Palace. It was built in 1892 by Sultan Muhammad Shah Aga Khan III. and has witnessed various vital incidents of the history, from being the rescuer of famine-stricken locals to the imprisonment of Mahatma Gandhi. This palace has seen a lot of dreadful incidents. 11th, at Labs Imagica. This amusement park has a lot to offer to an individual of every age group. There is breathtaking rides, water rides, snow park and various other light and sound shows. Twelfth, Saras Bagh. This place is apt to spend a lazy evening time chilling with the natural beauty of the city. You can also witness the temple which is located on the hillock near the place and view the setting of the sun. Thirteenth, Pataleshwar Cave Temple. Built in the 8th century, it is the temple which is hidden in the heart of the city. It has a Shiva shrine and the place is nestled between modern structures and developments. Fourteenth, Shindi Chatri. This place was built in the 18th century and is a memorial dedicated to a military leader, Maharaj Shindi. There are also photographs and paintings of the Shindi family. 15th, Osho Ashram. This ashram is situated inside the Koregaon Park and is well known for the gala time it gives to its guests. The ashram has a 40-acre medication resort which gives different types of treatments.
And on behalf of the Tilak Maharashtra Vidyapeeth Pune, again it's warm welcome to everyone. I would like to introduce about the ZSCT Thakur Shyamnarayan Degree College, Kandivali, East Mumbai. I am proud to say our college is affiliated to Mumbai University. The University of Mumbai is one of the oldest and the premier university of India. The University of Mumbai is one of the largest university in the world, and the university in 2013 had the 711 affiliated colleges. ZSCT Thakur Shyamnarayan Degree College was addressed in the academic year 2016 and is approved by the government of Maharashtra and affiliated to the Mumbai University. It fulfills all the norms and standards set by the University of Mumbai. Honorable Mr. V.K. Singh, Chairman, Thakur Educational Trust, Mumbai, and respected Mr. Rajesh Singh, CEO, ZSCT Thakur Shyamnarayan Degree College, Mumbai, both are the backbone of our institute. ZSCT Thakur Shyamnarayan Degree College provides extensive collegiate and co-curriculum experiences for the student, host of activities in which the students learn leadership skill, expand their academic horizons, and celebrate the cultural diversity. As reflected in the mission statement, TSDC will provide the benchmark career goal with the high level of excellence and the fostering education that is accessible, affordable, and innovative. At present, Thakur Shyamnarayan Degree College are running following 11 courses in a bachelor degrees. There are named as Science, Arts, and Commerce.
या कुन्देन्दु तुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना
Now I welcome and introduce the Tilak Maharashtra Vidyapeeth, Pune, which is taking effort to organize this conference, and it's my great time to talk about the few words of this university. Tilak Maharashtra Vidyapeeth is a deemed university in Pune, Maharashtra, India. It was established in 1921 at the suggestion of Mahatma Gandhi and named after the Indian independence movement activist Lokmanya Gangadhar Tilak. Tilak Maharashtra Vidyapeeth is a private college and affiliated by the UGC and accredited by the AICTE NAC. Tilak Maharashtra Vidyapeeth offers 90 courses in science and paramedical, medical, engineering, commerce, and banking streams. Popular courses are MSW, A&M, MPhil, BJMC, and MBA. Now, motto of this Vidyapeeth, Vidya Bodhakari Yashaha Sukhakari Vidya Gurunam Guruha. That is, knowledge is the secret treasury and beststower of success and happiness. It is the principal teacher, that is, teacher of all the teachers. Honorable Dr. Tilak, officiating Vice Chancellor Tilak Maharashtra Vidyapeeth, who gave an approval of organizing international conference in association. This conference is jointly organized with the next organization is Microbiologist Society India. It's my privilege to introduce the Microbiology Society India. Microbiologist Society was established in March 1996 and registered in November 1996 in Satara, Maharashtra, India. Any person graduate in a life science and interested in a microbiology can enroll a number of microbiologist society. Aim and objective of this microbiologist society to organize the courses in microbiology, to cultivate the license between the governmental and the non-governmental institution, to advise the government and its agencies on the microbiological problem of public interest, then cooperate and affiliate with the international organization in the field of microbiology, to encourage and support in microbiology, to establish the library of microbiology, etc. Welcome all. Now we start our conference with the pride of our nation and our national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
This today great event is organized in Maharashtra. I would like to pay our respect to our state Maharashtra with Maharashtra Git.
शिवरायांच्या पदस्पर्शाने पावन झालेल्या महाराष्ट्राचा तसेच कड्या कपारीने आणि त्यातून वाहणाऱ्या नद्यांनी नटलेल्या महाराष्ट्राचा गौरव करताना मावळ्यांच्या पोलादी मनगटाने मिळवून दिलेल्या स्फूर्तीतून शब्द उमटतात ते हे.
जय जय महाराष्ट्र माझा गरजा महाराष्ट्र माझा जय जय महाराष्ट्र माझा गरजा महाराष्ट्र माझा भीमा वरदा कृष्ण कोयना भद्रा गोदावरी एकपणाचे भरती पाणी मातीच्या घागरी हिमथडीच्या तटाराया हिमथडीच्या तटाराया यमुनेचे पाणी माझा जय महाराष्ट्र माझा जय जय महाराष्ट्र माझा गरजा महाराष्ट्र माझा. भीतीन आम्हा तुझी मुळी ही गडगडणाऱ्या नभा आसमानाच्या सुलतानीला जबाब देती जिभा सह्याद्रीचा सिंह गर्जतो सह्याद्रीचा सिंह गर्जतो शिवशंभुराला दरी दरीतून नाद गुंजला महाराष्ट्र माझा जय जय महाराष्ट्र माझा गरजा महाराष्ट्र माझा. काळ्या छातीवरी कोरली अभिमानाची लेणी पोलादी मनगटे खेळती खेळ जीव घेणी दारिद्र्याच्या उन्हात शिजला निढळाच्या घामाने भिजला देश गौरवासाठी झिजला देश गौरवासाठी झिजला दिल्लीचे हित राखी महाराष्ट्र माझा
Our institute is affiliated with the Mumbai University. I would like to present Mumbai University song.
सुन्दरं मन्दिरं शारदाया मुम्बापुरी विश्वविद्यालयम् इदं सुन्दरं मन्दिरं शारदाया मुम्बापुरी विश्वविद्यालयम् कलाशास्त्रवाणिज्यशाखाभिरूढा अमूढा विमुक्ता विहङ्गा वयम् शृण्वन्तु सर्वे दृढं निश्चयम् शृण्वन्तु सर्वे दृढं निश्चयम् इदं सुन्दरं मन्दिरं शारदाया मुम्बापुरी विश्वविद्यालयम् सत्यं वदामो धर्मं चरामो नयामो नृणा दुःखभारं लयम् सत्यं वदामो धर्मं चरामो नयामो नृणा दुःखभारं लयम् स्वकार्ये रतानां सदा जागृतानां भवेत् किं भविष्येति तस्मात् भयम् शृण्वन्तु सर्वे दृढं निश्चयम् शृण्वन्तु सर्वे दृढं निश्चयम् इदं सुन्दरं मन्दिरं शारदाया मुम्बापुरी विश्वविद्यालयम् युवा स्यात् बलिष्ठो युवा स्यात् गरिष्ठो युवा ध्येयनिष्ठो विभूयाद्वयम् युवा स्यात् बलिष्ठो युवा स्यात् गरिष्ठो युवा ध्येयनिष्ठो विभूयाद्वयम् यदि स्यात् युवा राष्ट्रकर्तव्यनिष्ठा समानेकतत्त्वं विलोकत्रयम् शृण्वन्तु सर्वे दृढं निश्चयम् शृण्वन्तु सर्वे दृढं निश्चयम् इदं सुन्दरं मन्दिरं शारदाया मुम्बापुरी विश्वविद्यालयम् वन्दामहे भारतं पूजनीयं स्वदेशात् परं नास्ति देवालयम् वन्दामहे भारतं पूजनीयं स्वदेशात् परं नास्ति देवालयम् अत्रैव सर्वे प्रतिष्ठापयामो ममत्वेन सार्धं समत्वं वयम् शृण्वन्तु सर्वे दृढं निश्चयम् शृण्वन्तु सर्वे दृढं निश्चयम् इदं सुन्दरं मन्दिरं शारदाया मुम्बापुरी विश्वविद्यालयम् कलाशास्त्रवाणिज्यशाखाभिरूढा अमूढा विमुक्ता विहङ्गा वयम् शृण्वन्तु सर्वे दृढं निश्चयम् शृण्वन्तु सर्वे दृढं निश्चयम् शृण्वन्तु सर्वे दृढं निश्चयम् शृण्वन्तु सर्वे दृढं निश्चयम्
Welcome back, everyone. It's time to inauguration of International Conference A very good morning to one and all with a great joy and immense exultation. I feel privileged to welcome our respected Principal Sir Dr. Sharat Pulari. We feel honored to have with us. Sir, you have hardly made any introduction. You have made all of us proud of your distinguished work in the numerous capacities. Transitions are never easy in any other practical stage of life. Principal, who is the man of the great, have nurtured and taught the students and have put their energy into making the college culture. You are the one of the different and respective dignitaries. Now I take the opportunity to introduce the wise and prominent personality, Dr. Sharat Pulari, sir. He has 21 years teaching experience as an associate professor, 8 years experience as a principal. He organized 33 conferences, 4 research projects completed under his guidance, 8 books and 85 papers published in a different national and international journal, experience as an NSS program officer and cultural coordinator. He awarded as the best teacher and the best college by Mumbai University. Dr. Sharat Pulari, sir, the man of the distinct vision and the fountainhead of illuminating ideas, an idol of knowledge and experience, and inspiration to all of us. Our honorable principal will address the inauguration of great event. I am confident that that speech will not just be collection of the words, but a strong motivational speech. So, we eager to hear you.
Good morning to everyone. On behalf of all the IQAC and Microbiology Department of Thakur Shyamnarayan Degree College, Andheri, Mumbai, and everything in between the Tilak Maharashtra Vidyapeeth, Pune, as well as Microbiologist Society, India, I stand here to offer you a warm welcome for International Conference on Study of Microbiomes today on 10th February 2020. So once again I repeat to all of you, very good morning to everyone. On behalf of the IQAC and the Microbiology Department of the Thakur Shyamnarayan Degree College, Kandivali, and the everything in between the Tilak Maharashtra Vidyapeeth, Pune, as well as the Microbiology Society of India, I stand here to offer a warm welcome for international conference on the study of microbiomes. That is today on the 10th February 2020. Warm welcome to the research delegates. Very warm welcome to the participants. Very warm welcome to the teacher colleagues, dear students, and the conveners, the organization committee secretaries, members. Of course, very warm welcome to the guest of honors, Honorable Dr. Tilak Madam, Vice Chancellor of the Tilak Maharashtra Vidyapeeth, Pune. Welcome to Dr. Pratik Dhawan sir, keynote speaker, and Dr. Hemant Tripathi sir as a honorable chief guest for the today's international conference with main theme as study of microbiomes. Of course, the warm welcome to the Professor Dr. A.M. Deshmukh sir in capacity of President of the Microbiology Society India. The main theme of today's international conference is study of microbiomes. It leads to research and review discussion, presentation about the microorganisms in a particular environment. The vision, mission and objective of this international conference is research and review about the microbiomes that protect us against the germs, breakdown of the food, that protect us against the germs, breakdown of the food to release the energy and produces the vitamins. It leads to research and review discussion at international level about the microbiome of the air, microbiome of the water, soil microbiome, industrial microbiome, food microbiomes, dairy microbiomes, microbiomes in medicinal plant and the interaction between the microbiomes and the plants. It is thought churning process about the microbiomes of the various places will be carried out in this international conference. Its advance are the research and the reviews. So dear friends, dear colleagues and the honorable guest, this international conference is for the study of the microbiomes, and it leads to research and review the discussion, presentation about the microorganisms in a particular environment. So really, as I said, it's an advance for the research and the review process in this field. For today's international conference, I feel fortunate to have Honorable Dr. Geetali Tilak Madam, officiating Vice Chancellor of the Tilak Maharashtra Vidyapeeth Pune. Honorable Vice Chancellor Ma'am means strong woman with a challenge in the eyes. Madam Vice Chancellor is iconic lady. Madam Vice Chancellor never limit herself. She never limits the other. Honorable Dr. Tilak Madam is a strong woman with a voice. By definition, she is strong in academics, but in the education to run the esteemed university that is Tilak Maharashtra Vidyapeeth. Great warm welcome to Madam Dr. Geetali Tilak, officiating Vice Chancellor of the Tilak Maharashtra Vidyapeeth once again. For today's international conference, I feel great fortunate to have Dr. Pratik Dhawan sir as keynote speaker. It's my privilege once again to announce that for the today's international conference, I feel very fortunate to have the Dr. Pratik Dhawan sir as keynote speaker. Honorable Dr. Dhawan sir is working as a postdoctoral research associate in neuroscience, Department of California, State University, United States. So, Sir Honorable Dr. Pratik Dhawan is working as a postdoctoral research associate in neuroscience department of California State University in the United States. It is great privilege to have Honorable Dr. Pratik Dhawan sir as keynote speaker. His excellence as a researcher in the neuroscience is a pleasure to listen with horse's mouth. It is one of the finest authority in field of research of the neuroscience. Warm welcome to the Dr. Pratik Dhawan sir for this international conference. We are glad to attend Honorable Dr. Hemant Tripathi sir as chief guest for this international conference on the study of the microbiomes. Honorable Dr. Hemant Tripathi sir is currently working as a researcher in biological science at African agricultural ecology in South Africa. I repeat, we are glad to attend Honorable Dr. Hemant Tripathi sir as chief guest for this international conference on the study of microbiomes. Honorable Dr. Hemant Tripathi sir is currently working as a researcher in the biological science at African agricultural ecology in South Africa. I would like to compliment to Honorable Dr. Hemant Tripathi for his most stimulating work as actual researcher person in the biological science. These points are to be underlined, and that's how I can say in the repetition. I would like to compliment to Honorable Dr. Hemant Tripathi sir for his most stimulating work as actual researcher person in the biological science. Execution of the research studies of the microbiomes is one of the finest expertise of Honorable Dr. Hemant Tripathi. Today, Dr. Hemant Tripathi sir will definitely share his great success stories about the studies in the microbiomes in the African agricultural ecology. I am confident about Dr. Hemant Tripathi sir's presentation for today's international conference. It will draw the audience in and will hold their attention for next technical session for this international conference. For the organization of the today's international conference, Thakur Shyamnarayan Degree College, Kandivali, Mumbai, must say that lot of thanks to Shri V.K. Singh sir, Chairman of Thakur Education Trust, and Shri Rajesh Singh sir, Chief Executive Officer of the Thakur Shyamnarayan Degree College, Mumbai. Shri V.K. Singh sir and Shri Rajesh Singh sir both are the personalities who support this college, who govern this college at par excellence. Shri V.K. Singh sir and Shri Rajesh Singh sir are ultimate supporters. Their support is solicited and acknowledged for their dedication toward this college. It's my moral duty to underline and bold these lines by the repetition. For the organization of the today's international conference, Thakur Shyamnarayan Degree College, Kandivali, Mumbai, must say lot of the thanks to Shri V.K. Singh sir, who is the chairman of the Thakur Education Trust, and Shri Rajesh Singh sir, who is the chief executive officer of this Thakur Shyamnarayan Degree College, Mumbai. Of course, I would like to say that they are great supporters to this institute. Their support is solicited and acknowledged for their dedication towards the college. Problems becomes an opportunity when the right people join together. Similarly, Udayvans Yadav sir of this college is working as a coordinator of microbiology in this college, and Mr. Ajit Khadilkar sir of the Tilak Maharashtra Vidyapeeth, Pune, is working as a secretary of the Tilak Maharashtra Vidyapeeth, Pune. These are the right people. They overcome the problems. They convert the problems into opportunities. And this opportunity is nothing but today's function, that is International Conference on the Studies of the Microbiomes. So this is how the capability of Udayban Yadav sir as well as Ajit Kadilkar sir, that they convert the every problems into opportunity, and this opportunity is participation of the researchers, delegates, students, the keynote presenter, and everyone in this international conference. Further, I must mention that the great things are done by series of the small things brought together. I repeat, the great things are done by series of the small things brought together. Similarly, this international conference on studies of the microbiomes, Dr. Bharti Wadekar Madam of this college carried out a series of the small things to achieve the great thing in the form of the international conference. I must to give the due credit, I would like to repeat, Dr. Bharti Wadekar Madam of this college carried out a series of the small things to achieve the great thing in the form of the international conference which is going to held today. Instead of to stand apart from the competition, Udayban Yadav sir, Ajit Khadilkar sir, and Dr. Bharti Wadekar Madam, three of these stand together as a team. Three cheers to this team and their efforts. When a team of the dedicated individual makes a commitment to act as one, then the sky is the limit for the success of the achievement. So, this is well said by the Henry Ford. Henry Ford says, when a team of dedicated individuals makes a commitment to act as one, then The sky is the limit for the success and the achievement. For the success and achievement of the today's international conference, organizing secretaries of this college, especially it is worth to mention about Sonali Joshi Madam, Kunal Thakur Sir, Aishwarya Kumar Madam, and Kiran Prajapati Madam form a team of dedicated individuals with committed act. I must have to say that for this international conference, organizing secretaries of this college to special mention Sonali Joshi Madam, Kunal Thakur Sir, Aishwarya Kumar Madam, Kiran Prajapati Madam form a team of dedicated individuals for the committed act, and this committed act comes out as a success and achievement of today's international conference organization. It is fact that in the right formation, the lifting power of many wings can achieve twice the distance of any bird flying alone. It is fact that in the right formation, the lifting power of many wings can achieve twice the distance of any bird flying alone. In conclusion, I would like to say, in the right formation, the lifting power of many wings extended by the organizing secretary, convener, co-convener, chief guest, keynote speaker, guest of honor, patrons, the research delegates, my dear student participants. These are the many wings. They come together to achieve the twice the distance of any one flying alone. It's amazing to welcome to everyone for this amazing international conference on the study of microbiomes. Thanks to everyone. Wishing a great success for a day ahead for the international conference. Thank you very much.
Thank you, sir, for your valuable words. Good morning, everyone. On behalf of the ZSCTS, Thakur Shyamnarayan Degree College, Mumbai, I welcome Dr. M. Deshmukh, who is the President of Microbiologist Society India. He is working towards connecting your minds, connecting young minds to strive towards research, success, and achievement. We are grateful to him for jointly organizing this event. Sir doesn't require an introduction as he is well recognized figure in our India and other countries. He is the President, Microbiologist Society India, former Chairman, Professor and Head Department of Microbiology, Dr. Babasaheb Ambedkar Marathwada University, Sub-Center, Usmanabad. He has 37 years experience in teaching and research, 82 papers published in national and international journals, 17 students have awarded PhD and one is working under his guidance. One major, two minor research projects completed, 4 books published and 7 books edited. He organized 45 state, national, international level conferences, seminar, workshop in India and abroad. He worked as a member of BOS, RRC, BUTR, environmental committees of many universities and the government. He worked as a resource person in 234 conferences, delivered the talk in 49 colleges and universities abroad. His talk, 14 talks on radio regarding the topic awareness was created regarding biofertilizer, biopesticides, pure water and vermicompost. 20 scientific articles on biotechnology were published in Marathi. He organized international conferences at Karad, Aurangabad, Sri Lanka, Bhutan, Mauritius, Dubai, USA, Andaman, Maldives, Nepal, Bangladesh. He conducting virtual classes in microbiology from the last 5 years. His interest in research area are nanotechnology, biotechnological solutions to e-waste, biodiversity of actinomycetes, biogas, and dye degradation. We are eager to listen your inaugural address. Sir, over to you for your great talk.
I am very happy to know today that Thakur Shyamnarayan Degree College Kandivali, Tilak Maharashtra University Pune and Microbiologist Society India jointly organizing e-conference on the topic study of microbiome. Now, microbiome is taking utmost importance in the world. The reason is that we require other sciences for the development of the human being. But microorganisms study is required to check the existence of human being. Because without study of microorganism, without knowing the activities of microorganism, human being existence is in danger. You take any field in the world related with the human being, agriculture, environment, transport, medical, and many other fields. In all these fields, microbiology is a very, very important. You see, which problems we are tackling today, whole world is tackling today, the major problem is in agriculture, chemical pesticides and health hazard. So, who can solve these problems? Microorganisms, by producing biofertilizer, means we can develop biofertilizer, biopesticide, these are nothing but microorganisms. And these microorganisms solve the chemical pollution caused by the chemical pesticides and chemical fertilizers. So in the agriculture, microorganisms are playing very, very important role. Microorganisms are eco-friendly tool in developing the agriculture. Then second, in the environment. In the environment, whole world is facing a problem of pollution. Which type of pollution? There is a solid pollution, there is a liquid pollution, MSW called as a municipal solid waste, biomedical waste, hazardous waste, and electronic waste. Like that, many other wastes are causing nuisance to the human being. Human life existence is in danger due to the development of human being, and as a result of the development, the pollution caused by the activities of the human being. One more pollution is a plastic pollution. Plastic pollution, government is trying to reduce the plastic pollution. Now everyone is asking me, what is an alternative? Again, microbiology is an alternative. How you see? To handle the electronic pollution, and we will say to create wealth from the waste, bio-leaching by microorganism can help to tackle the electronic waste. Again, municipal solid waste can be treated with the biodegradable microorganism. Liquid waste coming through the sewerage system is also treated with the microorganism. And for plastic waste, as it is created, we suggest to use the bioplastic. Bioplastic is produced with the help of microorganism, that is polylactic acid or polybeta hydroxy butyric acid. They are producing plastic. So, for environmental problem, the process is called as a bioremediation, bioaugmentation, biostimulation. So, in this field also, the microorganism, study of microorganism is a very, very important. Then let us come to the food microbiology. In the food microbiology, microorganisms play very, very important role. How? Again, I will tell you. Any food material which is a tasty, having good smell, good aroma, is produced by microorganism, produced by fermentation of microorganism. Number of examples I would like to give. Curd, whey, and pickle, bread. In addition to that, jalebi, khaman dokla, idli, and many others. All these are produced by the activities of microorganisms. So, in the food microbiology, microorganisms play important role. Even in the long storage of the food material, spoilage will take place only by microorganism. Study of microorganism will help us to preserve the food material for longer time. In addition to that, single cell protein and probiotics. These are the two important fields in the food microbiology. You know that in India, we have the problem of malnourishment. Every year, UNICEF is telling, every year more than 7 lakh children below age 5 are dying because of the malnutrition. How to solve the malnutrition problem? SCP, single cell protein, and probiotics are the solutions. Now, these food materials are available. These food supplements are available in the medical shop. You can purchase it from the medical shop. So, they supply large amount of protein, all vitamins, all amino acids, and all other required substances to the body, human body, are supplied by these microorganisms. So, the study of microorganisms, that is the study of probiotics, study of prebiotics, study of postbiotics, and study of single cell protein is a very important aspect for the microbiologist. Now, in the medical problem, maybe plant pathology and or maybe human diseases. In both the cases, study of microorganism is very important because there is a huge loss in the crop productivity if the plant pathogenic organism cause diseases to the produced crop or plant. Then human medical microbiology. Today is the best example. COVID-19, that is a coronavirus. Because one single tiny virus has stopped all activities of human being throughout the world. We are compelled to sit in the houses because of microorganism. So, medical microbiology is very important. On one side, these viruses are causing problem, but on another side, the solution is given by the viruses, that is vaccine. So, vaccine is a solution, and study of microorganism for the development of vaccine is very important. In addition to that, there are many other pathogenic organisms like a typhoid disease, like a tuberculosis disease, like a malaria, like a pneumonia, and hundreds of other microorganisms are there. So, study of all these microorganisms is playing very important role. So, as I have told, in every field now the study of microorganism is indispensable. That's why I congratulate the organizer for selecting the correct theme, which is a need of the time, which is a need of the nation, and all microbiologists have gathered today in this virtually, in this conference, should interact with each other, should ask the question to the speaker, and should get satisfied regarding the role of microorganism in the development of human being. In addition to that, I want to tell you, take any technology, prefix a bio word to it, it is a biotechnology. Today, bio-cement is available, bio-steel is available, bio-batteries are available, biosensors are available. In addition to that, bioinformatics, biofertilizer, biopesticide, bio-leaching, bio-bleaching. Like that, I can use about 100 different words where there is a importance of microbiology or biotechnology in the development of the human being. So, at the last, I want to congratulate and I want to appreciate the deliberations in this e-conference will be definitely useful, will be definitely inspiring and update the knowledge of youngster, because youngsters are very important in India. The population of youngster is highest in India. If they get educated themselves and they become ambassadors, in what sense? Today, all attending young generation will get the knowledge in this e-conference. They should translate, they should transfer this knowledge to the common people, to the lay people, or your family member, your neighbors, or village persons. Then and then only the awareness regarding microbiology will be created and our health will be safe, our problems will be solved, and we will be more healthy, more happy if we study microorganism. Thank you. Thank you again for giving me an opportunity to interact with the students in this e-conference. I am more thankful to Dr. Bharati Wadekar. She is constantly in touch with me for today's lecture. I am thankful to all. I am thankful to the management, principal, directors, and all authorities in this of these two universities for arranging today's event. Thank you. Thank you very much.
Thank you, sir, for your valuable talk.
Good afternoon, everyone. Now we start with plenary session. For this, I would like to invite Mr. Kunal Thakur for bio-note of keynote speaker Dr. Pratik Dhawan.
Thank you, ma'am. Very good morning to all. It is my great pleasure and honor to introduce you all with Dr. Pratik Dhawan this morning. I am thrilled as we have the opportunity to hear from Dr. Pratik Dhawan about the current situation in gene cloning. Sir has completed his PhD in life science from University of Mumbai in January 2021. During his doctorate, he has worked on various projects. He has experience in animal tissue culturing, animal handling, and virus culture. He has worked for Multidisciplinary Research Unit, MRC, in KEM, where he has gained knowledge in handling HPLC, GC, MS, PCR, and many more instruments. He is also working on various projects. Some of the projects are self-funded projects like electrospun nanofibers to evaluate wound healing activity in diabetic rats. And the other one is involvement of coated oxides nanoparticle involving any projects in Drosophila melanogaster against Parkinson's disorders progression. When he faced the challenge, he succeeded in utilizing different strategy in conducting these experiments. In his research career, he has published 14 papers in peer-reviewed journals and filed 4 national patents, out of which 1 is in nanoparticle synthesis and 3 in novel organic molecules. He also possesses brief knowledge of the in silico modeling of small molecules with a targeted protein. During COVID-19 pandemic, he has also volunteered for COVID-19 testing in Department of Virology, Hopkins University in Mumbai. In guidance of Dr. Sandipan Mukherjee, he is a person with immense knowledge and various field experience. He is currently pursuing his postdoctorate at Molecular, Cellular and Development Biological Department, Neuroscience Research Institute and MCDP, University of California, Santa Barbara. He has become an example of hard work and dedication for many of like us. Without wasting our time, I would like to request our guest of honor to deliver his speech so that we will able to gain new knowledge on gene cloning. Thank you. Now it's over to you, sir.
Good morning, everyone. My name is Dr. Pratik Dhawan. I am a postdoctoral employee at Molecular Cellular Developmental Biology, Neuroscience Department, California State University, Santa Barbara. Today, I would like to talk with you about genetic engineering. So, when we say genetic engineering, what are we trying to do? We are trying to manipulate or modify genes for betterment of human society, maybe. It's my privilege, and let me walk you through the basic principle of genetic engineering using CRISPR-Cas9. So basically, CRISPR-Cas9 is it's a bacterial mechanism, immune mechanism, defense mechanism, and we are using their defense mechanism into a state-of-art genetic manipulation tool. Before going into CRISPR-Cas, I would like you to tell about cloning, and because cloning is a base of CRISPR. Okay, so when we say genetic engineering, and what the first thing come in our mind? Suppose we have an electronic game, and there are multiple players playing that electronic game, and if you want to be ahead of them, uh, will that be great? Yes, why not? Everyone wants to be ahead of everyone, right? We want to be the first. So what if I could say that you have that game and you know the coding program of that game, and you can easily modulate or modify those codes according to your wish so that you will be ahead of that game. As simple as that, right? Exactly what we are doing in genetic engineering. We are just modifying or modulating some nucleotides in a DNA, or we are knocking in the when I say knocking in or knocking out, it's like deleting some nucleotides or adding some nucleotides. So when we say deleting some nucleotides, it is knocking out. And if you are adding some nucleotides or we are adding some genes inside a whole genome of an organism, we are adding gene of interest, what we are interested in. We are adding a different part of a gene, some gene, into a different genome of an organism. Then we call it as knocking in. Okay, so this is how we modulate or modify genes according to our wish for betterment of human mankind or human society, right? This sounds amazing. It's very amazing. It's like you have everything on your fingertips if you know CRISPR, if you know cloning. Okay, if you could manipulate genes in such a way that you can play with those genes. Say for example, I'm working with mosquitoes, okay, and what if I could say, if I can make a transgenic breeds of mosquito by manipulating their genes in such a way that in future those mosquitoes won't be able to see you. Will it be fascinating? Yes, it is. Then, as you all know, those are big nuisance to human society. They spread variety of diseases, and I think in near future that those insecticides what we are spreading, it won't work. And WHO has reported that the next pandemic might be because of those mosquitoes. So imagine you can manipulate those organisms which are dreadful to human mankind in such a way by modulating or changing their genes in such a way that those mosquitoes will no longer see you. They won't bite you, right? It is such a fascinating topic, right? So this is what a genetically modified organism, we call them as genetically modified organism. This is called as genetic modification or genetic manipulations. So basically, taking genes or identifying genes from organism, manipulating them, modifying them, and putting it back to that organism so that they can't be harmful for us, or we are improving their genes, right? We do not have any boundaries using CRISPR-Cas9. That is the beauty of beauty of biotechnology. That is the beauty of genetic engineering. Okay, so how will you do that? So first of all, we identify a gene where that gene is responsible for some protein which is expressing in that organism to to uh, 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 to say, for example, that mosquitoes. So I'm identifying the gene from opsins, that is rhodopsins, from the eyes of the mosquito. I'm mutating them, modulating them in such a way that they are no longer able to identify a specific wavelength of radiation coming out from human body, so that they cannot detect it and they cannot bite us, right? So identifying a gene, isolating that gene from a whole genome, copying that genetic material of interest using recombinant DNA methods, or by artificially synthesizing or modulating that DNA, putting that DNA inside a vector. Here, when I say a vector, we are using a word vector for a plasmid, bacterial plasmid. I'll tell you why we are using a bacterial plasmid as a vector for inserting those DNA and using those plasmids as a vector to transfer that gene into a new organism. Okay, in a future slides. So isolating a gene, copying that genetic material or modifying the genetic material, artificially synthesizing the DNA, or using CRISPR-Cas9, and that modified DNA or that modified gene, we are incorporating that modified gene into a vector, that is a plasmid, and then transferring that plasmid in an organism using CRISPR-Cas9. I'm telling that plasmid and CRISPR to exactly take this part and incorporate this part into a whole genome of that organism. So basically, then what I'm trying to do is I'm splicing some part of a genome, some part of a gene from a genome of a mosquito, and I'm replacing that part with my modified gene. That is called genetic engineering, because I want to know, once I hamper that gene, whether that protein will express or no, whether those mosquitoes will see me or no. Then I'll do the assays. Then I'll go forward. I'll check whether those proteins are expressing or no. So what I'm doing is I'm manipulating or knocking out or disrupting a gene in such a way that those genes will no longer make those proteins responsible for specific purpose. Then we call them a genetically modified organism. Okay, so this is how genetic engineering works. So the first step of genetic engineering is gene isolation. Then the second process is cloning and inserting a DNA into a host genome. Okay, so first we will go on to gene isolation. Okay, so basically what I'm doing, identifying a gene, cutting that gene into a using restriction enzymes. I'll tell what those restriction enzymes are in future slides. Or then amplifying that gene using polymerase chain reaction. Everyone might be aware of what PCR is. Okay, basically PCR is amplifying that gene into a multiple copies, so that those multiple copies can be incorporated into a plasmid, and then we can amplify those genes in a plasmid, in a bacteria, and then we could use those amplified gene into for transferring it into a host organism. Okay, so PCR basically makes multiple copy of that gene fragment from a whole genome. That is the use. That is the reason we are using polymerase chain reaction, PCR. After that, we should know that specific gene only has been amplified by using polymerase chain reaction or restriction enzymes. right? So then what we will do? We will do gel electrophoresis and cut that band or isolate that fragment, only a gene of interest which we want from that gel, and then we will purify that fragment from it, and then we will incorporate that fragment into a plasmid. Pretty simple process. Now, how will we identify genes, right? So basically, I don't. Everyone is aware of NCBI. If you are not, I would highly recommend you to go to NCBI, search for a gene, any gene, and there are some videos which are self-explanatory. Explore those videos and try to identify those genes, and try to use NCBI for PCR or making primers. Okay, so this is the page. Basic NCBI page. If I want to identify a gene, click here to a gene and just search it. Just go for it. Just search it, and you will identify. You will know at what gene or which gene is you want to modify or manipulate. So NCBI gives you a lot of options, and then once you identify the gene, you are going to amplify that gene using PCR. How will you do that? So PCR is for amplifying a gene, we need a specific oligonucleotide primers which are complementary. Those are short sequences which are complementary to that gene. So we have a forward sequence and we have a reverse sequence. Everyone knows gene amplifies using 5' to 3', right? So those primers are designed in such a way using primer blast in NCBI only, in such a way that they will attach to a gene specific specific site of a gene, and then they will amplify that gene. Okay, so that is this is how we amplify gene using PCR. Then once we have amplified a gene, then we will insert that gene, modified gene or manipulated gene into a plasmid. So everyone knows what is plasmid. I do not need to explain what plasmids are, right? So they have a different ORI sequence. They have resistant genes in their locus. So what we will do is we will cut plasmid using restriction fragments, restriction enzymes, and we will incorporate a different modified or our gene of interest into that plasmid. And once that is done, we will introduce this plasmid into a bacteria, so that we will get multiple copies of a plasmid, and thereby producing multiple copies of our gene of interest, right? So when I say you that we are using plasmids as vectors for amplifying our DNA, so basically they have unique restriction enzyme sites for cutting those plasmids and cutting those fragments. Okay, they are high. They have high transformation efficiency. They are easy to manipulate, and the purification process of plasmids is much easier than any other different mechanism. Okay, so that is the reason we are using plasmid. Okay, so this is how a basic plasmid skeleton is. So they have a ORI sequence, they have restriction enzyme sites, they have antibiotic. This is like antibiotic resistant gene. You can incorporate antibiotic resistant gene inside a plasmid, just like adding a different manipulated gene into a plasmid, exactly like that. So just identify where your restriction enzymes are, because restriction enzymes have a unique cutting site. Okay, they cut in a specific manner. Say for example, we have EcoRI. So EcoRI is a restriction enzyme. Restriction endonucleases will exactly cut in a specific manner in such a way that they will make a sticky ends. See, this the site of eco RI cutting sense, it is exactly opposite from upper strand and lower strand. See, this is how they cut it, because of which they make sticky ends. Once we are once we linearize the plasmid, I mean, once we have cut the plasmid, we call it digestion of plasmid, and your gene of interest with restriction enzymes, it is the time to link those together. Your plasmid, which has been cut now using restriction enzymes, and your gene of interest, which is a recombinant gene, you made it. You have to incorporate those to your gene of interest into the plasmid, right? How will we do that? See, this is the DNA backbone, which is a plasmid. We have cut the plasmid using restriction enzymes. This is the DNA insert, which is our gene of interest. We have cut the gene of interest using sticky ends, using restriction enzymes, and it also created a sticky ends using ecoRNA. The DNA ligase will stick them together, and we have a recombinant DNA incorporated into a plasmid. It is as simple as that. Okay. See, this is the flowchart of clone. This is the schematic diagram. So this is my gene of interest, which I have modified. I have cut. I have done the restriction digestion of that gene. Similarly, I have a plasmid where I want to incorporate this gene inside this plasmid. So I have digested it using ecoRNA and sal-I. The plasmid, the restriction endonucleases have produced a sticky ends. Then the DNA ligase will come and it will join the recombinant gene into this plasmid. Now, the question will come, why we have this ampicillin gene inside a plasmid? See, if I'm exposing my, if I'm culturing any bacteria on a agar plate, there are chances that there will be lot of contamination, right? I do not want that. I do not want that, because while screening, I need something that should give me an identification that only your plasmid containing bacteria have been grown on that plate. That plasmid has my gene of interest, right? So that is the reason we are incorporating ampicillin gene inside a plasmid that will behave as a marker. Right? So while plating, while making plates, agar plates, I will incorporate ampicillin in that agar and make plates. Since my constructed plasmid along with a recombinant DNA has a ampicillin resistant gene, only those bacteria which have plasmid incorporated my gene of interest and ampicillin resistant gene will only grow on that plate, right? See, only grow on that. Those bacteria will only grow on that plate, and then I can isolate those colonies only. Then I'll be sure that only those bacteria which have my plasmid of interest have grown on that bacteria. That is the reason we are incorporating ampicillin resistant gene inside the plasmid. Okay, so this is how we do the screening. Now, when we incorporate a plasmid inside a bacteria, it is called as transformation in bacteria, and we use stellar cells. Basically, those are bacteria only, E. coli strain, but those stellar cells are highly modified according to our will, so that they have improved cloning efficiencies. Okay, and they do not have any DNA containing irregular structures. Okay, so those stellar cells are basically provided by company. If you could order, they will you'll readily get those stellar cells, which are specifically made for transformation of plasmid. Okay, so they have those stellar cells are have various applications such as preparation of cDNA, genomic libraries, construction of longer length genomic libraries, or subcloning. So we are interested in cloning, right? So we are using those stellar cells for transformation of our plasmid, our reconstructed plasmid, which have our which have a recombinant gene, right? So we are using those bacteria, which are modified competent bacterial cell. We have a recombinant plasmid, which have a gene of our gene of interest, which is modified gene, recombinant gene. And now it is the time to transfer this plasmid into a bacteria so that the bacteria will make multiple copies of this plasmid. And indirectly, those plasmid will make in multiple copies of my gene of interest, right? How will we do that? So once we have a competent bacterial cell, those bacterial cells, we keep it on ice for bacterial cells. We introduce plasmid inside those bacterial cells, and we keep those bacterial cells along with plasmid inside ice bath for at least 30 minutes. So what happens, because those cells and plasmid are on ice, those plasmid will stick on the upper surface or the outer surface of the bacteria on ice. Once that is done, we give them a brief heat shock, exactly 45 seconds and 42 degrees Celsius. Exactly 45 seconds to at 42 degrees Celsius, we give them brief heat shock to facilitate the DNA uptake. So what happens is, because of the heat shock, the bacterial cell, the pores of the bacterial cells get open, and the plasmid which is sticked, which are stuck on the upper coat of the bacteria, will get incorporated inside the inside the bacteria. This is how we transfer our recombinant plasmid along which have our gene inside a bacteria. Once that is done, we transform that bacteria into a ampicillin LB medium plate. Okay, then those bacteria will grow on grow on this ampicillin LB medium LB agar plate, and then we can inoculate those. We can isolate those colonies, and then we can inoculate those bacteria into a LB broth, so that we can culture multiple copies of those bacteria, and then we can isolate the plasmid from those bacteria. Then we could do plasmid digestion, or then we could isolate that gene for incorporation that gene into a different organism. This is as simple as that. This the cloning is very simple. This is the whole protocol of cloning. It is simple. It is just tricky. Once you get a hang on of it, you will be able to do cloning in your lab. This is very simple. Okay, so basically what cloning is, I'll sum up. You identify a gene which you want to modify or manipulate. You incorporate that gene into a plasmid, into a vector, preparation of vector DNA, then to be cloned, creation of recombinant DNA, introduction of recombinant DNA into a host organism. Then we could make different copies of those plasmid, and then we could identify those plasmids using PCR or DNA sequencing, right? Once that is done, now you have a plasmid which have your gene of interest, your recombinant gene of interest, and now that plasmid has to be transferred into a host organism. Say for example, I have constructed a plasmid with recombinant gene inside it, and I want to transfer that plasmid into a mosquito. Okay, so what I will do, I will use artificially engineered techniques. So we have different techniques: meganucleases, zinc finger nucleases, transcription activator like effector nucleases, and CRISPR-Cas9. So today I won't go in detail of those things. I'll just give you a brief idea how in our lab how we use CRISPR guided RNA system for genetic manipulation. Okay, so what is basically CRISPR is? So CRISPR is made up of two components, a Cas9 enzyme and a guide RNA. Okay, so what are they basically? These CRISPR is called as clustered regularly interspaced short palindromic repeats. Basically, before going to CRISPR, I would like to tell you the mechanism of CRISPR is, what the mechanism of CRISPR is. Say, for example, bacteria. We have our own immune system, right? So once we are infected with bacteria or once we are infected with viruses, our body recognizes it for a longer period of time, right? So what is happening is once we have or once we are infected with bacteria and viruses, our B cells or T cells, so our B memory cells will recognize those bacteria or viruses, and once we when we are again infected with the same virus or a bacteria, my B cells will recognize them and then they will kill them. So this is our basic immune system, right? Exactly like this, a CRISPR is immune system of bacteria. See, once a virus invades a bacteria, it injects its genetic material, which is a foreign DNA. What bacteria will do, it will cut a fragment of the DNA and it will incorporate that fragment into its its chromosome. Okay, its DNA. So how they do it is, once we have a CRISPR locus and Cas9 gene producing Cas9 genes. So this Cas9 genes will produce Cas9 enzyme, and this CRISPR locus will incorporate those repeats, those spacers. Okay, so basically, so these are repeats of nucleotides. These are basically multiple nucleotide repeats, and we have a spacer here. So what happens is, when a foreign DNA comes in, bacteria incorporates this DNA into multiple spacers. Okay, so every time a new virus comes in, infects a bacteria, these spacers will increase, right? And these repeats will be similar. This is how bacteria know, because of those repeats, bacteria know where to incorporate a foreign DNA. Okay. So when a similar virus comes in, bacteria of CRISPR of bacteria will transcription will happen. They will transcribe RNA and they will make guide RNAs. These guide RNAs will identify the foreign DNA and it will give a double cut, okay, on in the host genome, and because of which the bacteria will survive. This is the survival mechanism of a bacteria. Because of that double cut, the genome of host will no longer be active and it will no longer transcribe itself or use the machinery of bacteria to make a multiple copies of it. Okay, this is how a bacteria survives. That is their defense mechanism, you can say. I hope you all understand what I'm trying to say. So basically, CRISPR is clustered regularly interspaced short palindromic repeats. So this is the repeat nucleotide. This is how bacteria knows where to incorporate a foreign DNA fragment of viruses inside a chromosome, inside their chromosome. So every spacer is a new fragment of viruses. So next time, if a bacteria, if a similar virus invades a bacteria, the bacteria will know that this virus has attacked him earlier, and it will cut the foreign DNA, and this is how the bacteria survives. See, this is the schematic diagram. Phage genes, it will incorporate some fragment into a CRISPR region, and when this same type of virus attacks a bacteria, the transcription will happen, guide RNAs will be synthesized, and these guide RNAs will have a complementary sequence of the gene of the host of the invading virus, and it will attach that complementary sequence to the invading gene, and it will cut the gene. This is how the mechanism of viruses stop there. This is the immune system of a bacteria. And now what we are doing is, as we all know now, this the CRISPR requires a guide RNA and a Cas9. So what we are doing, we are synthesizing a different we are synthesizing such type of guide RNAs of our gene of interest, which is complementary to the gene. So remember, we have incorporated our modified gene into a plasmid, right? Now, what we will do is, we will synthesize guide RNA in such a way that it will be a complementary part to a host genome, to the organism where I want to transfer that modified gene. So I will design a guide RNA so that once this guide RNA inside gets incorporated into the host genome, say for example, if I want to transfer a recombinant gene into a mosquito, I will design guide RNA in such a way that guide RNA, once it is incorporated into a mosquito, it will cut mosquito's genome exactly where I want it to cut, so that I can incorporate my modified genome there inside that fragment. Okay, this is how we design CRISPR RNAs or CRISPR genes, right? So as I told you, it's a mechanism of Cas9 and CRISPR RNA. So what are Cas9 is? These are nucleases, a protein that cuts nucleic acids exactly where I want it to be. So I'm making the use of bacterial defense system for my benefit, or modifying genes or incorporating genes into a host genome. So Cas9 will make a double strand break, and I will direct my manipulated gene or modified gene, which is inside a plasmid, now to get incorporated into a host genome, that is my mosquito or any other organism, right? So this is how a mechanism of CRISPR looks like. CRISPR-Cas9 looks like. So we have a guide RNA here. We have a PAM sequence here. We have nucleases which will cut a double-stranded DNA, and we have a Cas9 protein. Okay. So now the question comes: how a guide RNA will recognize a host DNA sequence where to cut? For that, guide RNA has a specific PAM sequence called as protospacer adjacent motif, or PAM for short. Remember, I told you, repeat and a spacer, repeat and a spacer. So when after transcription, when a guide RNA is synthesized, we have this loop. That loop comes from repeats. Okay, and this is a complementary strand, a complementary RNA of invading viruses, right? Remember this. Okay, so when I say how will I guide this RNA to cut exactly where I want it to be, it should recognize a PAM sequence from that repeats. Okay, so the guide RNA will recognize that PAM sequence. So the PAM sequence is we call it NGG, and N or the star is any nucleotide, but it should be linked with G and G. So NGG is a PAM sequence. The guide RNA will recognize this PAM sequence, and it will make exactly 3 nucleotides after PAM sequence a double stranded cut using nuclease enzymes. So see, so this is the host genome. This is the host genome. This is my Cas9. This is the linker loop or guide RNA. This will recognize a PAM sequence and it will exactly cut 3 nucleotides earlier the PAM sequence using 2 nucleases, HNH and RUVC. This is double stranded cut. Identifying PAM sequence. Once that is done, see, a double stranded cut is very much harmful for a cell because we are cutting a chromosome in such a way. So now the proteins will hamper, the cell won't survive, right? So cell's mechanism is it will either add different error-prone nucleotides inside it, or it will, which we call them as non-homologous end joining, or it will do a homology directed repair. Okay, so once a double stranded cut is there, cut has been introduced using guide RNA and Cas9, the cell will try to fill that gap. Okay, so if we are not doing anything, if you do not want to incorporate our gene, we just have to we just want to knock out a gene, I mean we just want to disrupt a gene, we will keep it as it is to fill the gaps by cell itself, right? So what the cell will do, it will rejoin that gap using non-homologous end joinings. This happens when this happens, a different nucleotides has been introduced inside the gene, inside that gene, because of this, what happens, the gene will no longer express a protein of interest which will enhance the which has some function, respective function. That is what we wanted, right? So if I want to make some deletions or make disruption in eyes of a mosquito, what I'll do, I'll make a double stranded cut using guide RNA and Cas9, and let the cell make changes using non-homologous end joining. So what the cell will do, it will try to join these two fragments of a gene using non-homologous end joining, so that it will incorporate some nucleotides inside it. But now the gene has been disrupted and it will no longer express a functional protein. If it is not expressing a functional protein, that protein is no longer be in use of my interest. So my mosquitoes will no longer see, right? This is how CRISPR-Cas9 works. On the other hand, if I want to incorporate a gene, which we have incorporated that gene into our plasmid, right? If you want to incorporate that gene, a recombinant gene, into a genome of a mosquito, what I'll do, I will do a homology directed repair. So this is say for example, this is a gene. Okay, this is a gene I want to incorporate inside a genome of a mosquito. What I'll do, I will, along with the gene, I will also incorporate some part of non-homologous genes into the plasmid, just beside a gene which I have modified. This we called as homologous arms. Okay, so once the double cut has been done using CRISPR-Cas9, what cell will do? Cell will try to identify this homologous arms. It will try to search whether there are any different genes available to identify whether these genes are matching these genes or no. So once cell identify these homologous arms, so now cell knows, yeah, this sequence is similar to this sequence, and this sequence is similar to this sequence. So this sequence is this, and this sequence is this. So now what cell will do? It will incorporate this whole sequence inside a genome of a genome of a mosquito. This is called as homology directed repair, because we call this homology because this is homologous sequences, right? These are homologous sequences, right? And cell is identifying these homologous sequences and then incorporating this recombinant gene, what I created, into a genome of a mosquito or genome of a host. This is how we insert a recombinant gene inside a host genome, or inside, say for example, in my case, we are incorporating this recombinant gene inside a mosquito genome by using homologous directed repair. So what I am doing exactly here is I am guiding this guide RNA to make a double stranded cut exactly where I want it to be, and by using homologous directed repair, I'm introducing this new gene which I created into the mosquito genome. Now this gene will express, and then I will know whether that is functioning properly or no. This is how I'm modulating or modifying a new species of mosquito that we call as GMOs, or transgenic mosquitoes, or transgenic breeds, for betterment of mine. So if I'm introducing new options or disrupting those options from eyes of a mosquito, which are expressing a protein, which is those proteins are recognizing a specific IR radiations coming out of my body, out of a human body. That gene, I'm silencing it, or I'm incorporating that gene, modified gene into that genome of a mosquito. And now that those options are no longer functional, so that the mosquito will no longer see me as a host. It will no longer bite me, and so the viruses, the different viruses, or the different viruses such as dengue or Zika, that won't be transmitting in humans, right? So I'm modifying mosquitoes, or I'm modifying organism for betterment of human mankind. This is how we do genetic engineering, using plasmid or using bacterial defense mechanism. Okay, so this is the schematic diagram. Cas9 guide RNA. This is the host genome. It will identify it. It will make a double stranded break, and then I will introduce my gene inside a genome, host genome, right? So here we call it, when if you are not incorporating anything, that is non-homologous end joining. If you are not incorporating anything, it is called as knockout or disruptive gene. We are not adding any specific gene inside. We are just cutting that gene so that that gene is no longer functional, or that gene will no longer make functional proteins. We are just cutting some nucleotides of it, right? We call it knockout or disruptive gene. If we are making adding some altered sequence or altered DNA, what we modified it according to our wish, then it is called as homologous and recombinant or homologous end joining, right? Homology directed repair. So this is how we do gene modulation or gene manipulation in organisms. So we have lot of scope in future for CRISPR-Cas9 mediated genome engineering. Say for example, I am making mosquitoes which are transgenic breeds, which no longer see humans in future. So we will be no longer be infected with dengue or Zika or any other viruses, Japanese encephalitis or any other diseases like that. because mosquitoes are host to host of the two different viruses, like, right, which are transmitted to humans. So I'm making them blind to humans. Just specific gene which are recognizing the IR radiations which are coming from humans. I'm just silencing those genes. So I'm making such kind of transgenic genes. And once I transgenic mosquitoes, and once I make those transgenic mosquitoes, I will introduce those transgenic mosquitoes into a wild, so that they can breed with a wild mosquitoes. They can transfer that manipulated gene inside a wild mosquitoes, and the progeny those wild mosquitoes will produce will have that modified gene of my interest, and they will no longer see humans as a host, right? This is how genetic engineering works. This is fascinating, and we have ample amount of future ahead of us by using this CRISPR-Cas9. And sky is the limit. If you know the basics of CRISPR-Cas9, if you could manipulate or modify genes according to your wish, you can do n number of experiments. You can find out how the genes work. You can find out the basic cascade, how the gene works. And you can modify those G protein coupled receptors. You can modify those proteins. You can check by silencing different genes how humans survive in extreme conditions. Or we could make we do not know, in future we could make a real Spider-Man or real Hulk in future. We do not know. CRISPR-Cas9 is like, we have n number of applications, right? This is how genetic engineering or genetic manipulation work. So I am working, as of now, I am working with working on a project which is funded by WHO, and I'm working to silence a gene specific to mosquitoes, which is like opsin gene. And we have an article coming up from our lab in New York Times, and shortly that article will come in Nature Communications, that what if we could become invisible to mosquitoes in future. So thank you so much for giving me this opportunity to share a basic knowledge of cloning and CRISPR-Cas9, what I know, or that can be helpful for you. And I hope you all you will be interested in driving such kind of genetic engineering tools in future. And if you have any doubts, you can call me, or you can mail me. So I'm really thankful to my my investigator Craig Montel, who gave me the opportunity to work on those fascinating cutting edge technology. And I'm also thankful for ZSCT's Thakur Shyamnarayan Degree College for giving me this opportunity to share my knowledge of cloning and CRISPR, and also to Mr. Kunal Thakur, who is an assistant professor there, and my it's a best friend, for giving me this opportunity. Thank you so much, and enjoy your day, and hope you all like this talk. Thank you so much.
Thank you, sir. Now, I would like to invite Mr. Udayban Yadav for Bionote of keynote speaker, Dr. Hemant Tripathi.
Thank you, Ms. Sonali Joshi, Madam. Good afternoon, one and all present over here for this international e-conference on microbiome, organized by Department of Microbiology, Interdisciplinary, Jhagdu Singh Charitable Trust, Thakur Shyamnarayan Degree College, Thakur Complex, Kandivali East, Mumbai, 4,101. On behalf of management, principal and faculties of Tsdc, I welcome our chief guest, Dr. Hemant Tripathi, Research Fellow at African Agricultural Ecology at University of Leeds, Edinburgh, Scotland, UK. About the sir, he is keenly interested in patterns, causes, changes, and consequences of local and global biodiversity. Sir have done studied on biodiversity change by observing the patterns within and between ecological communities. how species and functional traits at multiple scales, that is temporal or spatial, respond to different disturbances, that is land use, climate, herbivory, and others, and on how their responses underpin and affect functionality of ecosystem. Dr. Sir also interested in how species interact, that is complete or adapt in the changing landscapes as a result of the increasing human dominance and subsequent loss and fragmentation of original habitats. Understanding of biodiversity in a changing world is imperative for sustainable development. With a comprehensive understanding of pattern, response, and roles of biodiversity in ecosystem functioning and resilience, we can design the ecologically sound and biodiversity friendly landscape for future aspects. This is what the approach of Sir towards the nature. About the experience of Sir, Dr. Hemant Tripathi presently as an advisor on sustainability and environmental impact at DTIMA Energy Analytics, London, UK. At this moment, Sir is exploring ideas around the use of data science to forecast pest and diseases outbreak and develop early warning systems for farmers and other stakeholders. Sir has also experience in research fellow at African Agricultural Ecology, University of Leeds, UK. Here, Sir has worked with CGRF Africa project, which focuses on building resilient and sustainable agriculture system in Southern Africa. As a part of this project, Sir has investigated pest damage and control, ecosystem services and disservices, and biodiversity change in agriculture landscape of South Africa, Tanzania. Sir is a co-founder and ecology expert in. Sir is a co-founder of EcoBasics Ecology Consultancy Services, Mumbai, India. The main aim is to provide ecology and GIS related consultancy to profit and non-profit organizations in India. Also worked as a postdoctoral research at University of Edinburgh, Department of Geoscience, UK. Sir has experience on services for poverty elevations funded project, abrupt changes in ecosystem services, that is ACES. Sir has also experience in ecology as a biodiversity expert in Ecomen Laboratories, Lucknow. Sir is a research investigator, that is under the mango tree, where the role of indigenous beekeeping with Apis cerana indica in crop production in farm systems of Dharampur Taluka. He worked as a junior research fellowship in Shivaji University, Kolhapur. This is a DBT funded research project. Sir also worked on a assistant director on Beacon Television, where the content required for the science-based and wildlife-based educational videos program. Sir also worked on a executive program officer in Spandan Eco Foundation, where has organized a 10-day international youth summit on climate change and future sustainability, which inspiring a group of 60 international students from India, Netherlands, and Finland to surpass their differences and develop a united world view on environment and climate change. Sir also worked on a research officer on Bombay Natural History Society. And here he managed scientific publication databases on avian ecology and inland wetlands, and many more. With these many years of experience, sir, please enlighten our participants. Now I would like to request Ms. Sonali Joshi madam to take over the session. Thank you.
Hello and namaste, everybody. Warm welcome to all the participants, dignitaries, guest speakers, and also hosts and organizers of this wonderful conference. My name is Hemant Tripathi, and I am a postdoctoral research fellow in the University of Leeds. And a majority of my research is about understanding relationships between biodiversity, land use change, climate change, food security, etc., and also understanding how all these things impact human well-being and livelihoods and humanity and also global health. So I'm mainly going to talk about research relating to these topics, and also this is also the research that I've been part of in last 7 to 8 years of me being in the UK. So, but before getting into all the technical discussions or technical presentation, I can give you a brief personal background of myself. So I grew up in Mumbai, and I am an alumni of Thakur College. So I did my junior college as well as undergraduate degree from Thakur College, specializing in botany and horticulture. And I finished way back in 2005. So it's been a lot of time since then. And I think Thakur College has played a very important role in my journey and development as a researcher. I'm going to share all those things in the slide presentation through the slides. So I promise that my presentation is not going to be all the technical stuff, but also a bit of fun stuff. So I hope you'll enjoy it. Just a warning that I'm also going to use a bit of Hindi in between my sentences. So I hope you don't mind that. So without much further ado, let's jump on the presentation. So you can see on the slide that I'm mainly going to talk about biodiversity, food production, and climate change. This is the research that I've been part of in the last many years, from my PhD, which started in 2013, to my postdoctoral research, which started in 2018. These topics are also global challenges. So biodiversity loss, food insecurity, climate risks, and land use change. So land use change basically involves deforestation and degradation or rapid change of a land cover. So all these things are global problems. These problems are going to affect humanity in future. So I'm going to talk about these global change problems, and also how, as an ecologist, I'm contributing to solve these problems, to provide a solution for these problems to policymakers and various other stakeholders in the landscape. So I already mentioned that I'm part of University of Leeds, and I work there as a postdoctoral research fellow on a UK government-funded project called AfriCap. And this AfriCap is funded by another program called GCRF. GCRF is Global Challenges Research Funding. So they basically fund big global or regional level challenges. So I'm part of this GCRF funded project called AfriCap at the University of Leeds. Alongside that, I'm also a co-founder in a data science company called DTime AI. And DTime AI is basically a data science company that focuses on building data-driven tools to improve sustainability, and mainly sustainability in the energy and the agriculture sector. So I lead the agriculture aspect of DTime. This is the structure of today's talk. So I'll give you a very brief background of my journey from Mumbai to Leeds. And it also involves a lot of time in Africa, because a lot of my research is focused in Africa, in sub-Saharan Africa. So I'm going to also talk a bit around that. And then I'm going to broadly talk about biodiversity and land use change, some of the theoretical aspects that might give you a bit more knowledge about this problem. Then I'm going to discuss about a very new concept, or relatively new concept, called climate smart agriculture. I'm not sure if you've heard of it, but a lot of my research in the last 2 to 3 years have been focused on evaluating climate smart agriculture from the perspective of ecological implications. And then I'm going to briefly mention about some of the projects relevant to using big data in the D-Time company that I'm part of. And then I'm going to summarize whatever I've spoken, provide you key points, and also some brief conclusions on what being an ecologist means in a changing world. So I came to the UK in 2013, and before that I was in India and based out of Mumbai. I already mentioned that I did my undergrad in Thakur College. After that, I went to Siddharth College for a master's in environmental botany, all this journey started in 2002 when I was introduced to the wonderful world of plants and plant-animal interactions by my teachers in the Thakur College, mainly Vineet Vaidya and Sushil Shinde. So they played a very important role in introducing me to the nature, to the natural world, to taxonomy, to even photography. And so a lot of my time in Mumbai was spent exploring the Sanjay Gandhi National Park, a lot of areas in Western Ghats. Also, I was part of a filmmaking company which were making documentaries about the natural world and biodiversity. So I spent a lot of time with them exploring the length and breadth of India and doing a lot of interesting photography. So you can see these are some of the pictures that I clicked during that time. And the landscape that you see in the background, it is somewhere around Pune. So I spent a lot of time exploring landscapes and exploring botany and plant diversity before I came here. I also spent a lot of time teaching. So I was part of the Bombay Natural History Society as an education officer, and I led many nature trails. Then I also went to a lot of botany explorations. So in the bottom picture, the boy who's holding the camera trying to photograph some plant in the wetland, that's me, and that's somewhere in Sawantwadi. And then I also did a lot of wildlife photography. So you can see a beautiful moon moth that I've photographed somewhere in Gujarat, and then the wine snake. I think this picture is from the national park. So I spent a lot of time exploring wildlife, learning about the biodiversity in India, with a special focus on plant and animal interactions. And I think that helped me build a lot of knowledge, which played a very important role in me being where I am right now, I think. And in just around in 2010, 2012, I started a company in Mumbai called EcoBasics. And through EcoBasics, I was doing a lot of surveys of biodiversity and trees and mapping trees. And by 2013, I decided that it's enough of doing the same thing over and over again. And I should probably explore learning opportunities. And that's when I started applying for scholarships. Another project that I did in the North and Western Ghats in Gujarat, in the Dangs region, was about pollination ecology. So I was basically studying how beekeeping and the pollination due to the bees in the bee boxes, how that helps crop production. And that research played an extremely crucial role in making my applications for scholarships very strong. So through that research, I could show that I not only have a potential as a researcher, but also a potential as a more a researcher who can also investigate, explore social aspects, economic aspects. That highlighted my potential as an interdisciplinary research, and that made my profile very, very strong. So in 2012, I started applying for scholarships, and I fortunately got a fully funded PhD offer from the University of Edinburgh, which is one of the top 20 universities in the world. And I got a very prestigious scholarship called Principal Development Research Scholarship, and not many people get it, so I was thrilled. So in 2013, I moved to this beautiful city of Edinburgh, and in this picture you can see the Edinburgh town from a hillside. And I think this hill is called Arthur's Seat. So it's a very beautiful city, and Edinburgh played a very important role in developing me as a researcher. So when I was in Edinburgh, my research was more focused on understanding biodiversity and land use change in 3 countries in sub-Saharan Africa: Mozambique, Zambia, and some parts of Malawi. So, and I focused on a very beautiful landscape. So these trees that you see, these belong to a species called Mopane, and these in this region you'll only find Mopane trees, and that's the reason this whole landscape is called Mopane woodlands. So one of the most beautiful landscapes that I've ever been into. So I researched mainly in the Mopane, and there's another similar landscape called Miombo. So most of my research was in Mopane and Miombo woodlands, and I was studying biodiversity of all kinds. So I was sampling birds, sampling insects, sampling trees and grasses, as well as also sampling mammals, and I used a lot of different kind of techniques. So basically my PhD gave me this freedom to explore, to learn many different techniques, from sampling techniques to also statistical techniques, and that really helped me to build my profile as a researcher. So in this landscape, you can see this is the Mopane woodland in Zambia on the left-hand side. On the right-hand side, you see these are some elephants which are just coming out of a Miombo woodland. And this is a picture from Kruger National Park. And when we think of Africa, we think that it would be a very different landscape, but in reality, it has very similar composition, social and ecological composition like that of India. So the problems are the same. The wildlife is as exciting as Indian wildlife. So you can see this picture from one of my research sites in Mozambique, where I was investigating how abrupt changes in the woodlands affect well-being and livelihood of people. So most of my research is motivated by this idea that how does food production, biodiversity interact with each other? What are the trade-offs? What are the synergies? What are the factors driving the synergies or driving the trade-offs? So when I say trade-offs, I mean that when biodiversity and food production are against each other. For example, you can either do food production or save biodiversity. So a lot of areas around the world are into trade-off. So you can either conserve biodiversity or you can make food, but that's not how we can be sustainable. For being sustainable, we need to find synergies. We need to find landscapes where you can have a bit of food production, you can have a bit of biodiversity conservation, or you can have areas that are dedicated for biodiversity conservation and areas where you can do food production. So most of my research is trying to understand how we can improve synergies and reduce the trade-offs, right? So, but why are we interested in food production and biodiversity? So you can see that in this slide that the global land area, only 15 to 20% is forest area, and a lot of non-forest areas are the dominant land use across the globe. And so if you really want to save biodiversity at the global level, we need to focus on these non-forest, non-protected areas as well. And then also the global demand for agriculture is predicted to increase, because as human population is increasing, we will need more food and we need to produce more food. And when we need to produce more food, we can either produce food by intensifying food production. So by using pesticides or fertilizers, so intensify food production, you can get more food from a small area, or you can expand agriculture and increase your food production. So when you expand agriculture, that means that you need more area under agriculture. What does that mean? That means that we will probably remove the protected areas, remove the more natural areas, and convert them into agricultural fields and produce more food. So food production has to increase by at least 60% by 2050 to support our growing human population. And that will have some impact on biodiversity. So, our global food system is the primary driver of biodiversity loss, with the agriculture alone being the reason for over 50% of species going under risk of extinctions. And unless we change the way we produce food, this biodiversity loss will continue to accelerate. And as you can see in this map on the slide, which shows the projected effects of agriculture intensification, expansion, and both intensification as well as expansion on biodiversity, the blue shades indicate lower biodiversity losses, 10 to 20%, while the red represents biodiversity losses in the level of over 50% of species going locally extinct. So this map basically suggests that in future, if the increased food demand is met through conventional expansion and intensification of agriculture by clearing habitats or using chemical fertilizers and pesticides, food production will trade off strongly against biodiversity. And what did I say in the start? That we want to reduce these trade-offs, but actually these trade-offs is going to increase if we continue to produce, if you continue to consume the way we are doing right now. So we can also see in this map that the countries with the least suitable agricultural lands are also the countries with agriculture expansion potential are likely to be the ones hardest hit by increased food demands. So there's going to be a lot of pressure on biodiversity because of our increased demand in food. So, and then due to this pressure, there will be a lot of biodiversity loss. But then biodiversity loss also means that it will be very difficult for us to live without biodiversity. So it is widely known that agriculture has severe impacts on biodiversity, as I clearly mentioned in the previous slides. But biodiversity, when we lose a lot of biodiversity and the benefits that it provides, this biodiversity loss will also have a lot of negative impact on food production, because biodiversity produces or provides ecosystem services. So biodiversity provides pollination, pest control also. And through these services, it also improves the resilience of our food system. So if biodiversity loss is caused by food production, then it will also threaten the sustainability of food production itself. And a great example could be pest and pathogen control services provided by biodiversity via natural enemies. So about 20 to 40% of our annual yields are lost to pest and pathogens. And in some cases, almost whole crop field is destroyed during pest and disease outbreak. So natural enemies suppress pests and reduce the yield losses induced by pests and pathogens. So this is extremely important in small scale, low input systems where farmers rely on biocontrol and traditional management practices. So despite this recognized ecosystem services supplied by biodiversity to agriculture, the feedback loop of agriculture production impacts on biodiversity, and then biodiversity's impact on agriculture production is not often considered in planning of agriculture interventions. So bottom line, we need to increase food production, but this food production, if we increase the way we are doing right now, it will impact biodiversity. And if it impacts biodiversity, then because we have lost biodiversity, we will also lose some of the ecosystem services that supports food production. So it's a cycle. You increase food production, you lose biodiversity, but then when you lost biodiversity, you also reduce food production in the future. So we need to get out of this cyclical loop. or cynical loop. And then another major problem is climate change. So whatever we discussed before, we did not consider climate change. When you consider climate change in this formula, things become even more complicated, right? So climate change impacts agriculture by reducing the productivity and fertility of crops. So जब climate change होगा, तो हमारे crops की fertility और productivity दोनों increase होगी. So which means that we have to basically put more grains, have more area under production to basically produce the same amount of food that we need, right? So, and then it also affects cropping cycles, growing seasons, which consequently also reduce yields and increase the cost of production. So with climate change, we are expected to basically lose about 20 to 30% of future yields, right? So जो भी हमने increase किया हमारे crop yield by expanding agriculture or intensifying agriculture, all that, or at least 20% of that could be lost just because of climate change. So which means that पहले हमने सोचा था कि हमको 60% grow करना है, but हमको now 60% grow करना है, plus जो 20% climate change से lose होने वाला है, उसको भी हमको कहीं ना कहीं से adjust करना है. So it makes the whole situation complicated. Plus, climate change can also cause imbalances in interaction between species. So climate change will cause some local extinctions, amphibians के losses हो जाएंगे, and then a lot of biodiversity is also very, very sensitive to replacement. So a lot of generalists will take over. A lot of specialists will get locally extinct. And then because of climate change, there are also range shifts and many species. So when climate change is happening and the temperature is increasing, तो जो species lowland areas में रहते हैं, जहाँ पे already गर्मी है, for example, Mumbai, it's already hot. So when climate change happens and temperature is increasing, what will the animals do? Because for them, it's too difficult to live in a heating environment. So they might want to move. So they will move to higher altitudinal areas. So what will happen then? We'll lose a lot of species in the lowland. We'll have a lot of species in the highlands, and species which cannot migrate. So species which are not adaptable, they will die. They will get locally extinct. So we will get a lot of species on the highlands, almost no species in the lowlands, and that will have its own unique effect. And many scientists say that we do not even know about the potential impact of range shifts. One potential impact of range shifts could be that जहाँ पे पहले crop pest नहीं थे, वहाँ पे नए pest आएंगे, नए diseases आएंगे, नए livestock के diseases आएंगे, नए human के diseases आएंगे. So that will create its own problem. So right now we are seeing a human pandemic, but there will also be, and there have been crop pandemic, crop pest, livestock pandemics. So we will lose a lot of food because of these things as well. And then because of these shifts in biodiversity due to climate change, there'll be shifts in the services that biodiversity provides. So as I mentioned in the previous slides, that biodiversity provides pollination, biodiversity provides biocontrol of crop pests and diseases, and also human, in fact, the zoonotic diseases that impact humans are also basically controlled by biodiversity to some extent. So when we have these shifts in biodiversity, there will also be shifts in the services and disservices of that biodiversity provides. So disservices, I mean pests. Biodiversity also provides pests. So there will be pests in areas where we cannot even imagine, new areas. And when these pests go to new areas, we do not know how they will impact. People in that area are not prepared to deal with this impact. Governments are not prepared to deal with this new pest impact. So it will create its own problem. And all these will affect food production as well as biodiversity. So I mentioned before that we need to increase food production to feed the growing human population. And this increase in food production will come at the cost of environment and biodiversity. And we need to increase these food production by 60%. Then I told you that climate change की वजह से we lose 20% of the food we produce in future, 20 to 30%. And we need to also factor that in our plans. But now I'm saying that there will also be effect due to changes in shifts or shifts in services and disservices. So you can account additional 30%. So हमको 60% बढ़ाना है food production, but there are roadblocks, 20% by climate change and approximately 30% by the changes in crop pests and diseases. So there's a lot of problem. And then there's another factor. There's also international trade. So another major driver of food as well as biodiversity alongside climate change is international trade, which often also is associated with increase in commercial and mechanized agriculture. So जैसे कि जहाँ पे ज़्यादा trade होता है वहाँ पे हमको देखने मिलता है कि commercial agriculture और mechanized agriculture भी बढ़ गया है. And what this commercial and mechanized agriculture do is they inherently create more homogenized systems, because small scale agriculture में थोड़ा सा maize हो गया, थोड़ा सा wheat हो गया, थोड़ा सा oat हो गया, थोड़ा सा beans हो गया, but commercial agriculture, large scale agriculture में we have to make hectares of maize or hectares of wheat. So that causes homogenization of the landscape. And when there is homogenization of landscape, again, the same thing happens. We lose biodiversity. We lose the services provided by biodiversity, and due to which our system becomes vulnerable. We lose resilience. What is resilience? Resilience is the ability of the system to fight back. But when we have removed biodiversity, when we have lost biodiversity, our system, our food production system becomes weak. We become vulnerable. And so trade plays a very important role, because as we get more connected, trade does two things. It makes the landscapes more homogeneous, because trade generally demands only, let's say 12 to 13 products, commodities, and farmers have to make those, have to grow those commodities. So it homogenizes the landscape. Another thing that trade does is when we are moving from America to, when we are moving, for example, grains or something from Africa to America, or from China to Africa, we are also, there is a risk of transporting new pests and diseases to a new landscape. For example, in Africa, in East Africa, not many years ago, there was a new pest that was introduced through the market connectivity, which is called fall armyworm. And now fall armyworm is the biggest threat to the crop production in the region. When the fall armyworm outbreak happens, some farmers lose 60 to 100% of the crop yield. So again, so I mentioned you about 4 very interacting problems: food production, biodiversity loss, climate change, and trade. All these 4 interact with each other, and we need to find a way to understand synergies and trade-offs among these 4 very complex things. And that's what, so I do not do that whole research involving this whole big global changes, but my research connects these key drivers. And also, one other thing to mention is that biodiversity is context dependent. How biodiversity responds to change depends on which organism is involved. Where is this change taking place? What is the scale? Is this a 1 kilometer scale, 10 kilometer scale, or 100 kilometer scale? What are the management practices in the field? How by management practices I mean, what are the specific practices that farmers are deploying in the field, and what are the practices in the neighboring field, and then what is the landscape composition and configuration? All these things matter. So हमने जो चीज एक जगह स्टडी की है, हम उसको हर जगह replicate नहीं कर सकते. So we need to take into consideration many case studies when we are trying to understand this complex problem. So what we really need is, we need to design sustainable and resilient food systems for the future, and a food system that has capacity to cope with many shocks. And this system should be resilient and adaptable. So resilient means that it should be able to withstand a shock, and then it should be able to adapt to that shock. And in this, when I say the system, this system includes biodiversity, but it also includes people, because the people are making decisions to have biodiversity or have diversity in the landscape. And then these systems should be multifunctional, so they should provide multiple benefits. So when I say multiple benefits, food production is a benefit, but alongside food production, ability to save biodiversity is also a benefit. So it's also a benefit and a function. So this is multifunctional. So we should have multifunctional landscapes. Then there should be win-win support. There should be support for win-win services. So having biodiversity, having ecosystem services, still producing food, as well as fighting with climate change and maintaining sustainability. So this is win-win scenario. So we should focus on win-win things. And then it should contribute to biodiversity. So our landscapes should provide support for biodiversity. Biodiversity, we all think that biodiversity has to be only protected in the national parks, but that's not true. As I showed you before, only 20 or 25% of the global land surface is protected area. Rest all is non-protected. So we have to save biodiversity even in the non-protected agricultural landscapes as well. And then we have to link to the large-scale drivers. So large-scale drivers are basically the drivers that I mentioned earlier. So trade, climate change, these are large-scale drivers. So our system should have a link. Whatever we plan to do should have a link to the large-scale drivers. So to solve some of these problems to some extent, to help make our food systems more resilient, there's a new concept that have been introduced. Some of you might have heard about this concept, some of you may not. This is called climate smart agriculture. So climate smart agriculture is basically proposed as an approach to food production that can improve productivity, increase resilience, and also improve adaptation to climate change, to risks, and also decrease the agriculture's greenhouse gas emissions. So CSA can basically create more sustainable and resilient food systems. And then CSA means climate smart agriculture, also aims to support sustainable and equitable transitions for agriculture systems and livelihoods across multiple scales, ranging from smallholders to transnational coalitions. So CSA is proposed as a solution for some of these problems. So CSA is climate smart agriculture, but there are other relatives of CSA, like conservation agriculture or nature-based agriculture. So some of these concepts are introduced in India as well. So in Andhra Pradesh, I think Andhra Pradesh government introduced nature-based farming. In the sites where I did my research, the governments have introduced climate smart agriculture. So my research mainly focused on understanding what are the ecological implications of climate smart agriculture. Is it doing what it is supposed to do? What are the trade-offs? How are different people and biodiversity impacted? How is the soil impacted? So I was trying to basically look at the system, look at the whole landscape where CSA was introduced, to understand more about the pros and cons, the cost and benefits, etc. So I conducted this agroecology research in Tanzania, basically to evaluate CSA in that area. And CSA basically involved having a very specific set of spacing between plants, having agroforestry alongside crops, then doing more of crop rotations, intercropping, and then also include some trenches. So trenches, basically big trenches between two crop rows, and put the soil above the trench and plant trees on that trench, so that some of the trees on the side of the fields can provide shade, and the leaves of those trees can also provide improved soil nutrition. And this study, if you see a map of Africa, on the eastern side of Africa, there's a small country called Tanzania. So I did this research in there. So I basically evaluated CSA from an ecological and socio-ecological point of view. And basically, I conducted a year-long study, installed a lot of different kind of insect traps, so pitfall trap, sticky trap, and pan traps, to basically sample some insect species. I also spoke to the farmers to understand about their perspective, did visual essays of crop damage, And when I say I did, basically I had a team of people doing that for me. And I used to train and visit the sites frequently, train them and collect samples. I also worked with a lot of farmers who helped in conducting the research. And this is what the landscape looks like. So this is the East to Samvara landscape where I conducted this research. So it looks very pristine in nature. And this is the idea that we have in our minds, that nature should always mean very pristine and without people. But if you look carefully in this landscape, there are also villages. So, and that's why we call these systems socio-ecological systems. So there's a lot of ecology, but there's also a lot of, there's a social aspect to these systems. There are people in it. There's people, human well-being angle to these landscapes. So, and majority of global Earth surface, majority of our Earth contains socio-ecological systems. ऐसा कहीं नहीं है कि सिर्फ nature ही nature है और people नहीं है. It's a mix of nature and people, and these are the landscape that really need more understanding, right? So we were here working in this landscape, and this is exactly the place where we spend a lot of time. This is my research station in the East to Sambara Mountains, the tiny white house that you can see. Those are where our research stations, where we did a lot of work, and we had a lot of meetings. We put our samples there. And we basically worked with invertebrate or insect specimens largely. And one of the challenges that we faced was that not everybody was an insect taxonomist. Even I am not an insect taxonomist. I cannot identify insects. And this is a major challenge a lot of ecologists face, that they do not know how to classify and identify a lot of specimens they collect. But then I realized that we don't really need to identify and classify everything to the species level. Just understanding what are the different type of groups that are found in the landscape is enough. So here we were working with in the Tanzania site, we were working with the farmers who did not know much about insect identification or classification scientifically, but we still wanted to work with them. So what we did was we asked them not to bother naming these different specimens. They can just group them color-wise, size-wise, structure-wise, and just give them numbers or names they like, name in their local language. So we call them operational taxonomic units. So based on morphology. So we worked with farmers, trained them a bit, but then they helped us collect thousands of specimens and identify them to the operational taxonomic unit level so that we could do the study. So this also emphasizes that the value of collaborating with local communities. We cannot always bring scientists or experts to the field. We also need to collaborate with people and work with what we have. Then another study that I was part of is understanding how climate change is impacting agriculture and resilience in South Africa. So in South Africa, it's a very similar story, but there's a slightly different problem. In Tanzania, the major climate problem is a flash flood. So हर 3 या 4 महीने में there's a flash flood, there's a huge rainfall, and बहुत सारे crops and बहुत सारा landscape get destroyed in Tanzania. In South Africa, exactly opposite is happening. Almost every year, there's a delay in the rainfall, and there is a long dry period. So there's a drought in South Africa, and you can see in this chart that drought is a major problem. And the colors indicate the size of the farm. So in Tanzania, all the farmers are small scale. सबकी छोटी-छोटी fields हैं, almost half say 1 hectare की. While in South Africa, there are many different kind of farmers. There are large scale commercial farmers who have over 200 hectares of land. There are medium scale farmers who have 50 to 100 hectares of land, and there are small scale farmers who have less than 10 hectares of land. So we try to categorize these farmers and try to understand what challenges they are facing, and also try to understand that how these things are impacting them and what they are doing in return. So we understood that drought is a major problem in this landscape, and drought is mainly affecting livestock health. and leading to losses in yields due to crop failure, and also pest damage was a major problem. Some of the farmers could not plant in time because there's no water. They don't want to plant and let the small seed get dried and die. So they would delay in planting, and then eventually they will lose crop and even livestock sometimes. And then we try to understand what they are doing in response to it. And some of the farmers started practicing a concept of agriculture called conservation agriculture. So they started mixing their crops with cover crops, with other plants, to try to diversify farming, doing multiple crops. Some started to do genetically modified crops as well. So our goal then was to understand what is the impact of this transition? What is the impact of these adaptations? So this can be adaptation. Farmers are adapting in response to a shock. So our goal was try to understand how this adaptation, how this response to a risk, to a climate risk is impacting ecology, is impacting biodiversity, for example. So that was another research study in South Africa. And this is very close to my research station in South Africa, in a small province called Free State. And this is basically the landscape just behind this lake. There are some farms and fields where we worked. And South Africa is a fascinating country to work. So very close to a research site, there's also a national park where we usually sometimes go and see wildlife. So there's a beautiful male leopard staring at us in that area. Also, not very far from a research site, we go and we can see African elephants. There's a beautiful picture of two bull elephants fighting with each other for maybe the territory. And this is, again, one of my most favorite national parks. It's a Golden Gate National Park in South Africa. Again, very, very close to a research site. So based on our research studies in Tanzania and in South Africa, what we did was we tried to identify the practices which can have positive association with biodiversity. For example, we found that mulching and trenches and space and intercrop have a positive relationship with yields, but also positive relationship with biodiversity. And then we also found that maintaining a complexity, doing a lot of different things in a field helps improve resilience. So diversity improves resilience. And then we also found, if you see on the right-hand side of the slide, you can see these multiple pictures, scatter plots. So what is this essentially showing is that how biodiversity impacts food production or crop damage really depends on what kind of biodiversity is there in your field. So if you see in these plots carefully, you'll see red bands and blue bands and green bands. So green band is basically showing places where there was high richness, high diversity. So you see this green band, it is starting from high value and going to the lower value. So that basically means that when there is a lot of biodiversity in the landscape, then your crop damage reduces as biodiversity increases. So when there's a lot of biodiversity, there's less crop damage, which means there is more yield. So you can have better yield, especially when there is a climate risk, if you maintain some of the biodiversity in the field. For example, if you maintain natural enemies. So natural enemies means wasps and parasitoids, spiders, for example. If you maintain population of these key insects, then your crop yields can be better even when there is a climate risk, right? And then you can also see, but when you do not maintain this diversity, what happens? You can see that the red band indicates low diversity areas. So the red band in one of the plots, in the top left plot, you see the red band is starting from bottom and going up, which means that in a low diversity, the crop damage increases, while in again and annual yield, crop damage increases and annual yield that you get from the crops decreases. So which basically again means the same, that low diversity system, there is a huge risk for pest damage, but in a high diversity system, the risk from pest damage is greatly reduced. So that was a key point. And then in these areas, when the COVID happened, we also did a study to understand how COVID impacted the farming communities, which kind of farmers responded or survived best in the COVID-19 impact. You don't have to read all these things on the slide. I can just explain it to you. So basically what we found was, so we interviewed a lot of farmers in Tanzania, in South Africa, and we found that farmers who had a lot of diversity in the field. So when I say diversity in the field, I mean not only just biodiversity, but also crop diversity. So farmers who were growing maize, who were growing wheat, who were growing bananas, who were growing cassava, who were growing some cash crops like clove and spices, and also food crops. So maize and wheat are food crops. So farmers who were doing mix of food and cash crop and vegetables, they survived better, because COVID-19, what it did, it impacted the value chain. So वैल्यू चेन को इम्पैक्ट कर दिया. अब बाहर के फूड अंदर नहीं आ रहे, अंदर का फूड बाहर नहीं जा सकता है. So what the farmers who were dependent on the market, they could not survive. They lost a lot of money. They could not, they become very poor. They had a lot of food insecurity. They did not have money to even buy food. But farmers who cultivated mixed group of things, mixed range of things, they survived better, because okay, we cannot travel, and so हमारे spices अब market में नहीं जाएंगे, but at least we have maize, we have vegetables that we can survive on. So farmers who basically had very diversified fields, they were able to survive in the pandemic really well. So key lesson हमको क्या मिलता है? Key lesson is that diversity in the field improves your resilience, improves your adaptability, improves your ability to survive in a risky environment, and that is what we should promote in our agricultural systems. Abhi kya ho raha hai? Because of trade, because of global demand, commercial demand, हमारे जो agriculture systems हैं, their diversity is decreasing. So again, it's not just biodiversity. I also mean crop diversity. Hamari crop diversity is decreasing. If you go to North India, you'll see fields after fields, only wheat, only rice. So which basically is a problem. If there is a risk, then all that will be going to be wiped down. And COVID-19 is just one risk. There can be a climate shock, there can be a drought, there can be a flood, and there are a lot of problems about with flood in North India, for example. In Maharashtra, there's a problem with drought. So in a climate risky environment, we should always have very diversified farming conditions. So what did we learn? So one of my job is to basically try to estimate what the future looks like based on what we have observed, based on what we have studied. So हमने हमारी पूरी understanding को use करके Tanzania के लिए future में क्या होने वाला है उसका picture build किया, and we use this understanding to inform the government and introduce maybe new policies. So for example, we made this interesting tool called as an iFeed tool, and basically we showed our study in form of 4 quadrants. So you can see there are 4 quadrants, and basically this is a message to the government that if you do this, then this happens. So for example, in the present status, human capital matlab, in a human capital scenario, there is a technological stagnation, and there is a also very low climate risk. तो बेसिकली जैसा अभी है वैसा अगर सिचुएशन होगा तो बायोडाइवर्सिटी का रोल बहुत इंपॉर्टेंट है, राइट? बट देन अगर क्लाइमेट कैओस होगा, अगर बहुत सारा क्लाइमेट रिस्क होगा, तो क्या होगा टंजानिया में? सो वी पेंटेड अ पिक्चर फॉर देम दैट इफ देयर इज अ क्लाइमेट कैओस देन देयर विल बी इंक्रीज इन पेस्ट एट हाई ऑल्टीट्यूड्स. और तुम्हारे कितने प्रतिशत इकोनॉमी हाई ऑल्टीट्यूड से आती है? Let's say 5%, but that's a huge 5% is a big thing for a country. So we can say that अगर climate chaos होगा तो तुम्हारे high altitudes के area में इतना loss होगा. So you should focus on that, on those areas. Then what happens if there is a techno fix scenario. A techno-fixed scenario मतलब climate risk नहीं बढ़ेगा, risk नहीं बढ़ेगा, but there will be a high technological adoption, which means that all the farmers will become very techy. They will have access to a lot of instruments. They will have access to a lot of mechanization. So what will happen in that scenario? So we said in that scenario, what might happen is that your land will become extremely homogenized. So right now, Tanzania has very heterogeneous land, very diversified land. But if you adopt a lot of technology and not make people aware about the importance of landscape diversity, then what might happen is that there will be a decrease in biodiversity and pest control because of biodiversity. What will that mean? That will mean that larger fields may have high yields, but all that yield might be lost because of pest outbreak, because of high vulnerability to pest outbreaks. Then what happens if there is a scenario of intensive vulnerability? For example, in intensive vulnerability, there is an increase in climate risk, but there is very low technological adoption. So what that happens? So in that we say that there will be a severe increase in pest, and there will be also very simplified landscapes. So what is the solution to this? The solution is that understand what are the different scenarios, and based on that understanding, identify what are the tools that you have. So we produce something similar to the for many governments that we worked with, and we help them understand the climate change situation better from that way, and also based on our studies. So now that brings me to the D time aspect. So all of that I spoke before is the research that I did in being at the Leeds University, but I'm also part of a D time of a company called D time, which is a data science company. And in D time, what we are essentially trying to do is we are trying to create tools for data discovery. So, for example, you have probably Google to find all your data, but then Google usually gives you a lot of information, but it does not give you data sets. We are trying to build a tool that specifically helps you find data sets. And then we are also trying to build some tools that improve sustainability in energy and sustainability in the agriculture sector. My job basically involves looking after projects that improve sustainability in the agriculture sector. And one of the projects that we are, my team is right now working is, it's funded by a UK government through a grant called Innovate UK. So they fund all very innovative and exciting ideas through this grant. So one of these project is trying to make a statistical model or artificial intelligence based model of locust outbreak. So I don't know if you heard, but then in 2019, there was a locust outbreak, which started in East Africa, but it eventually also came to Pakistan and India. So you must have heard there is locust. I think in Hindi it's called tiddi. So there were a lot of locusts in India, but these in India as well. But then in Africa, these locusts caused massive problems. They devastated a lot of crop fields, ruined a lot of crops. Many farmers lost crops, and big organizations like World Bank and FAO, the way they were handling this locust outbreak situation was they were spraying pesticides on top of the locust swarm. So locust ki jhund pe, they were basically spraying pesticides, and spray of these pesticides will have environmental as well as public health implications, right? So what we are trying to do now is we are trying to put together all the possible locust data sets that are there in existence. So we have, for now, we have collated about 44,000 points of locust occurrences. And we are planning to model all of this, like put in a statistical model, make some machine learning, artificial intelligence algorithm, so that we are able to predict in advance where this locust outbreak can happen. And once a locust outbreak happens, in which direction and how they're going to move, so that farmers and organizations can know about a locust outbreak at least a couple of months in advance. And we are also aiming to do something similar for all the major crop pests and diseases. Why we are targeting crop pests and diseases? You will be surprised that crop pests and diseases, they are responsible for at least 30 to 40% of losses in crop yields. So they are a major problem. So we are basically trying to solve that problem. So what are the key take-home messages from my talk? So, all of my research is essentially about trying to evaluate agriculture transition, agriculture technologies, or new ideas in agriculture. So we evaluate them from the point of view, from the perspective of ecology. We try to understand what are the ecological impacts. So we understand, or we highlight, our work highlights that all the agriculture policies and related activities, they have to be evaluated always. Every few years, they have to be evaluated for their impacts on ecology as well as society or human well-being. Then these evaluations should look beyond productivity. मतलब हमारा एजेंडा सिर्फ इतना नहीं होना चाहिए कि फूड प्रोडक्शन हो रहा है कि नहीं हो रहा है. We should look at food system from multiple perspectives as an ecologist. Then we need more local scale studies. So there are a lot of global studies. हमारे बहुत सारे data जो हैं वो global है. हम India level पे study कर देते हैं, and we say that, oh, broadly this is what is happening in India, but that is not helpful. We need local scale studies. We need to know what is happening, for example, in Borivali, and what is happening in Karchat, for example. So we need more local scale studies so that we can give some guidance based on those studies. Then we also learned that diversity is very important for resilience. We saw where I showed you examples of how diversity in the farm, diversity in the field helped some of the farmers survive better in the pandemic, in the COVID-19. And then also broad level biodiversity is also very helpful in building resilience of the whole food system, because it provides ecosystem services like pollination and pest control. And then big data can solve a lot of problems. Big data can solve agriculture problems. It can also inform management of landscape, forest management, etc. So we need to use big data. We need to access and harness big data to solve some of the big problems. So as an ecologist in a changing world, we need to use multiple techniques. We need to be good at a lot of things, not be best in it, but at least be good in it, at least have some understanding. So as an ecologist, for example, if you take my own example, I trained as a botanist, but I did my PhD in geography, and right now I'm doing very interdisciplinary things. I'm using big data as well as collaborating with people who are doing artificial intelligence, collaborating with ecologists as well as biodiversity scientists to try and learn something about the system. So my one-line message would be, do not restrict yourself to one single domain. Always explore, become very interdisciplinary and multidisciplinary and more problem-focused. Like majority of my work is very problem-focused. In my case, the problem is trying to understand sustainability of the agriculture system. And this sustainability can be assessed from multiple ways. And that's exactly what I'm trying to do. So more problem-facing approach than a domain-facing approach is needed to solve global problems. So with this, I would like to end my talk. It's been a long talk for 55 minutes. I hope I haven't bored you, and I hope this has been interesting. I'll leave this presentation with this slide. So on the slide, you can see my contact details, my email ID for both the organizations that I'm part of, the D-Time and the University of Leeds. So feel free to reach out to me if you have any questions. Also, if you want to just share your ideas, if you want to communicate with me for collaborations. Also, if you want to do any summer internship, any projects which are related to my research area, then I'm happy to chat with you. Thank you very much. Have a very, very good day.
First part of session is completed. Now it's time for lunch. We will meet after half hour. Thank you. Thank you all.